Greetings, my dear students, and have a nice day too. And today's session, I am going to explain you how to model a protein by using online server, especially Swiss Modeler. And it is going to be in a completely online, so you need not worry about the softwares and the codes of to model a protein. It is less time consuming. For that, you go to the Google and give the keyword of Swiss model or Swiss modeler or Swiss workspace like that. So for Swiss protein modeling, you can give any keyword for that. It will redirect to you for the website that is called swissmodel.xpassi.org, and that in the home page you can find the start modeling. Right, this tab, just click it. You can get the workspace where you can put your FASTA sequence or protein sequence in this space for model. Okay, importantly, in the documentation tab, you can find excellent video tutorial which is already provided by the technical person of Swiss model. So if you have any doubt, you can go there and watch those small videos. They have covered all the nook and corners of the procedure and the results interpretation, all those things. And here what I'm going to do is I am just going to sum up how to perform the model, a quick summary and a quick hands-on also for you. If you have, what I'm telling is, if you have any doubts apart from this means, you can just go there. That is a very good tutorial available on the website itself. Okay, the input supported formats are given here. If you have a sequence, you can give. If you have an alignment file, you can give, and otherwise you can use a predefined template as a PDB file. Then DPWare project is also available. In this case, for the example, I'm going to model a protein called P2Y2, pyronergic receptor type 2Y. And so for that, we need a sequence. I will prefer UniProt to get the protein sequence. You can go NCBI also, right? So P2, Y2, and just you can give your own protein name, full name, or protein code, whatever is available. Then you will get, I'm going to model a human protein, and I just check the length of amino acids. For this, you can use NCBI to confirm the length of amino acids, length of protein. Okay, and here I will choose 366 and 377 is there, pyronergic receptor 2. I will choose here. Okay, and directly I need a sequence. I will get sequence from the left hand panel. So just download, and I can just copy from this page, and you can just paste in the given area. Right, if the sequence is okay means it will give the color lines like that. So if have any error means it will show you. Sometimes non-proteins or non-amino acids will be there in the sequence. Okay, you can give the project title, whatever you need. I have given pure energetic for the as a title, and you can find two options, and one is build model and another is search for templates. If you use build model option, you can just go there and click, which means all the steps will be done by Swiss model on the behalf of you, and it will produce the final result with the top 3 or top 2 models within 15 minutes or less than 15 minutes. But you don't know how it was happened, right? So for that you can use classical way, and this is called search for templates. This is the first step, right? We need templates to model, right? This is homology modeling. For that we need templates, right? Usually we do BLAST. This is what going to happen here. If you click search for templates, it will run like that. Okay, so it is searching for the templates, what are available templates in the databases or resources. So it will take few minutes to complete. So the alignment process is over. We have got 50 templates. All the templates are aligned, arranged by using the coverage score, and this QM GMQE is the score, global mean quality estimation score is most important thing. You have to, by using this score only, you have to select the best models. Okay, and that QE score identity, a method of uh, protein identifier, and oligo status. Mostly, not mostly, you should not select if any oligo state available in the protein, means you should use monomers, not oligomers for model. And the information of ligands, if any, means you can get from this column. Okay. Apart from this, you can go for quaternary structures available of the oligomeric states. You need not to do that. Stoichiometry. If you use anything, if the related proteins available, means you can get the green color line here. In this case, there is no green color line found in this monomer and the dimer, so you can go for the resolution. Okay, then, and this is about protein interaction fingerprint. Most of the proteins you can get 2 or 3 monomer, dimer, and oligomers like that, and in this case you get only one, a homodimer. um, the score should be negative, right? And the thing is, what is that? Interface conservation, and here is sequence identity. Sequence identity we can see, and interface conservation means how many amino acids are and which part of the amino acids may be mutated or it's prone to mutate. And it's negative means it's not get mutated. Okay, so the score should be in negative and the line should be in the negative, less than 0. Okay, this is about unsequenced similarity. These are the clusters you can choose among the 50 templates, and the clusters are available with similar structures. You can choose clusters also for model clusters as a template to model a protein. And here alignment of the target and the selected one. Okay, coming to the template, we have to select for model the protein, and this is P2Y2 receptor. Somebody has already modeled by using models of AlphaFold database model, and we cannot rely on that. We can use, okay, and according to the GMQE score, you can go for 0.59. So we have to select, and this also 0.59, and this also 0.59. Okay, this 0.57. We can select a top scored sequences templates and the query coverage, and unfortunately we don't have any full query coverage templates available here except the AlphaFold model, that is modeled one. This is the only one available with 100 queries sequence coverage, but it is modeled by someone, right? And these are the available in maybe available in PD because X-ray crystallography with resolution. So where I was, we have to select template, right? So for this 100% query coverage is there, but it is modeled protein already deposited by others. We cannot rely on this. We have to uh, choose other next three with the score highest score by 0.59. So totally 4 templates are selected to build a model, right? I already told you there is no other sequences query with 100% available for this protein. In your case, you may get 100% queries coverage. For that you can use as a template. Usually it will be in the first two, right? You can use. And next, template are selected. The next step is just click the build models. So the model will be developed. So the 4 models can be developed by using that template which we have already given. So model 3, and this is transmembrane protein. So this is the cell membrane, this outer or inner space of the protein. And this is model 3 with a score of 0.55. So global mean quality estimation score is most important to choose a good model. So this is 0.55. So most probably it is 0 to 1. The highest value will be good, right? So highest value means more than 0.7, 0.6, at least 0.6 will be better. And sequence similarity, sequence identity is 37.84. For this, that QME score is available. So you have to check this. As a result, you can take a snapshot of this and you can structure as assessment. You can go for assessment. For that, you will get the Ramachandran plot. By this way, you can get the score of that Ramachandran plot also, right? And see this. So the score is Ramachandran forward resistance is 94. Outliners, deviations, and bad angles, clash score will be 2%, 1.1%, 1% like that. So the better model should get above 98 percentage of score. It will be the best. Above 95, okay. If it is 98, means that it's very good model. Sometimes you can get 98%, more than 98%, which means that model is very good, best model. Okay, here let's see score, and this is outliners, and this is the stability of the amino acids. Here Q means score. We will see in the main page. Okay, this model 3, model 1. The model 1 will be best always, but model 4 and model 2 is there. According to the score, they have a lineup that 0.55 and here is 0.79, but this is by what is it, alpha fold database model. Okay, that's why we omit this and we can go for and this thing. It has the Q score of 0.55. The another one has the Q score of 0.56 and here is 0.56. Okay, the coverage is 37.84. Then here the coverage is about 37.84. So we can choose this will be a best model. So you have to check the quality of the model. So by producing the result of this thing, see that predicted local similarity to target this, right? And here you can go for Q mean Z score. This is most important thing to evaluate the model, whether your build model is good or bad. See, this is Q mean score, and this is beta carbon angle bonds, how much clashes will be there, like that. This is the, all atoms, then solvation, whether the protein is soluble or not. This is not soluble. And torsion, the angle of rotation of the each atoms of the protein will be negative. And here, and this is the important one, we can compare our model with the PDB structures, X-ray crystallography structures, and here and these are the total structures available in the PDB, and this black color has the Z score less than 1, which means normal distribution less than 1 deviation will be there, so don't worry about those calculations. If our model, this is our model position in this graph, if this star is in this position means in the block line means your model is good, right? So here the model is not that much good because the deviation is there, it is away from the block line and the block dots the area, so it is not that much good model. You can select for another one as a structurally you can select for another one. So as per this results, we can go for model 1, but this is model 1. You can get the 100% sequence query, 0.79 of global mean quality estimation score, and but thing is there is no QSE scores available, Z scores available. See that this is alignment alone. So for this purpose I omit this. I can take this model 3 and the overall score is 6.5, right? 6.6 is there. One more model 2. For model 2 you can and see this is that score, and here also same, right? If your model is very good means this star will be placed in the region dark, anywhere in the dark region. Okay, that means it has less stability, it has less if the star is in the block line means it has more stable, that is the thing, more stable structurally, and it possesses all the quality of the stable structure. Okay, then you can take screenshot of this and can produce the result, and you should take the Q mean Z score screenshot also, and you need this outliners also, predicted local similarity to targets, and it should be above the blue are good, right? And it should be above 0.6. If it reaches the 0.6 and below means it is not a good structure. In this we can get 1 2 3 4 5 6 7 places they can get the number of, see that local quality estimation chain A, it has 0.6 is okay, up to 0.6 is okay. In this structure there are more outliners, 0.5, right? Residues number, how many residues we take, somewhat beyond 300 residues, more than 300 residues. So the thing is, if the residues are above 0.6 means that structure is good, right? And here there is a line 0.68 like that. So these are all the good, these amino acids will be positioned in the good places, so those are more stable quality estimation. And here is very less, predict a local similarity to the target is less. Okay, now you can produce the best models PDB by downloading this PDB format, and you can show in PyMod or any other visualization software. Okay, so you can rotate and you can take PNG snapshot, and if you want to use anything in here you can go for site settings, you can go for Cypred prediction of secondary structure, VSP you can use, and color you can give colors for alphabetic, where is that? So you can use colors also, already in color you can give that. Okay, so you can show that. Let's just can go to default. and this is confidence gradient. In this picture we can explain these brown colors are not that much confidence, so the blue color are well structures, it has good structure stability. Okay, this is all about SWISS model. You can submit the PDF file as a result and some supporting result documents to your model evaluation. Okay, we will see in the next session.